Today, it's my pleasure to have Dr. Amit Bar-Or as my guest. He's an expert on multiple sclerosis, and we're going to learn from him current understanding of what might be going on in the central nervous system that leads to the clinical problems. And this is one disorder in contrast to some of the disorders I've worked on, Alzheimer's and Parkinson's, where there's actually treatments that have been developed that help patients a lot. So he'll talk about that. He's currently the Melissa and Paul Anderson President's Distinguished Professor and Chief of the Multiple Sclerosis Division in the Department of Neurology at the University of Pennsylvania School of Medicine. He's also the director of the Center for Neuroinflammation and Neurotherapeutics there. Amit, could you talk about, before you get into the science, where you grew up and how you got interested in medicine and science in general?
Sure, Mark. Thanks for having me today. So I'm born actually in Jerusalem, Israel, moved back and forth with my family largely because of my father's work. He was a clinician scientist, actually in the field of pediatric exercise physiology. We lived in Canada, in the US, in the UK, and back and forth in Israel. Over the years, I must say that I am one of those sort of nerdy growing up kids who decided he wanted to go into medicine from very early days. So it wasn't really a question for me as to whether to go into medicine or not, but then the question was what to do. And I was fascinated with the mind, the brain, like many others, especially when things go wrong, and figuring that must be one of the most challenging experiences for an individual to live with a condition that affects the central nervous system. And at the time also, as I was going through residency, I knew I wanted to do neurology, but there I had the experience of working with a few terrific mentors, Peter Riskind, one of them, who was also a clinician researcher and specialized in MS. So I had the chance to see how he interacted with, how he tried to better understand and help people living with this condition, a condition that affects, as you know, people relatively younger than some of the conditions you just mentioned, so that they face living with this condition for many years, for decades, during the time that they're trying to build families and vocational responsibilities and so on. So the sense was that if one could impact this condition, it may have a substantial impact for many years on people and their families. And at the time, there was also the beginning of the explosion in the fields of both immunology and neurobiology. So as someone who's interested in the academic aspect as well, it seemed like this amazing interface between two worlds that were just on such an exciting sort of development trajectory.
The two most complicated systems.
Well, yes,
in many ways.
Yes, I agree. But as you pointed out, it was also the time that MS had transitioned not that long before from yet another diagnosable but not treatable neurological condition to one that was, with treatments that for the first time changed the course of MS, but modestly. So there was a sense of excitement that it can be done, but there was also a sense that more needs to be done.
Yeah. And so you went to medical school in Canada, and then you did your residency and fellowship at Harvard Medical School. Is that right?
That's right. Yeah, I was at McGill in Montreal for med school, and then Boston for 7 years.
So you first were exposed to multiple sclerosis in Boston then?
Well, I had some experience in neuroimmunology already in research as a medical student. I did, the typical sort of medical student research rotations. But then really it was at the Mass General there that I was exposed to MS in a more serious, meaningful way. And then I continued with my fellowship at the Brigham and Women in the Center for Neurologic Diseases there with Howard Weiner, David Hafler, Samia Khoury, and others. So it was a great background to have.
Yeah, so you did an MD and essentially postdoc then at Harvard, where you got into the basic science and you learned about some of the tools you can use and okay, and then so can you talk about if a patient comes to the clinic and is having some neurological problems, and how does one diagnose multiple sclerosis?
So our approach to diagnosis has evolved over the years. What we've known for many years, for decades, is that in an individual who's living with MS, most individuals will end up with a relapsing remitting onset of their MS, right? So these are individuals who will experience some constellation of symptoms that come and then settle and then often go away, often completely on their own, or they may leave someone with a degree of new deficit that becomes their new baseline. And that is a relapse and remission. And about 85% of individuals with MS have that kind of a course. And there aren't that many things that can do this neurologically over time, with episodes coming and going and lasting weeks at a time. But there are a few, and certainly when someone presents for the first time and you don't yet know whether it's MS or not, there are several other conditions that can mimic MS. And so the diagnosis historically has required there to be, as the name would suggest, multiple sclerosis. There has to be involvement in more than one region of the central nervous system, and there has to be evolution over time. And the third, which people don't always think of as part of the diagnostic criteria, is that you need to exclude other things that can mimic MS, because there is a short list of other things. And all the more important nowadays to have the correct diagnosis, because some of these other conditions will benefit from different treatments and even sometimes may be made worse by treatment for one disease as opposed to the correct one. Excuse me. So the diagnostics were improved substantially with the advent of the MRI, because the MRI allows us to see the multiple spots quite readily, and that allows us to meet the criteria both of the multiple lesions in space, but also as you follow the MRI, or if you see lesions of different ages, some of which enhanced with the contrast that you might infuse, the gadolinium infusion, for example, and others that do not, those also allow you to fulfill the criteria of multiplicity and time. So the MRI has been very, very helpful. And we have been moving more and more towards trying to diagnose and offer treatments earlier in the course of MS.
And the damage, the lesions are seen both in the brain and spinal cord?
Yes, the MS involves the central nervous system, which is made up of both the brain and the spinal cord. And pathways in any of those regions may be involved, and hence the broad range of symptoms that people may experience because of these types of involvement of inflammation in the spinal cord and brain.
And you mentioned that usually the onset is in fairly early in life and not in older people. Are there any risk factors, or, it's an autoimmune disorder, and typically we think of women as being more susceptible to autoimmune disorders. Is that true for MS? Well,
it's true for many autoimmune conditions. There are a short list of autoimmune conditions where there's actually a male predilection, but for the most part, there is a female predilection, and MS is one of them. As you point out, most typically, MS will present in people clinically for the first time in their maybe 20s, 30s, and 40s. But there is also pediatric onset MS, an area that has been of particular interest to us. It's considerably less common, but it does happen. And in fact, we have an inkling that the development of MS biologically probably starts many years before the first clinical symptoms become apparent, and may well involve pediatric or adolescent age groups. There are very interesting epidemiological studies where people have tried to understand MS in populations. It's been well known that MS is not just randomly distributed around the globe. There are areas of higher risk and lower risk. North America is a relatively higher risk area. And you can ask the question as an epidemiologist as to what happens to the risk if you happen to migrate from an area of high risk to low risk, or vice versa. And the interesting answer in the context of MS is that it depends on the age at which you emigrate. So if you emigrate before, likely early adolescence, you will acquire the risk of the place you go to. But if you emigrate after adolescence, it looks like you bring with you the risk that was in the place where you were raised. So there's likely to be an important exposure that plays on top of the genetic contribution. So the risk factors that you're referring to that we think may play during this window of risk in relatively early life include things like a certain genetic predisposition, although the genetics of MS are not simple genetics. They actually have discovered to date that there are over 250 or so variants of the genes that can contribute. Not a single one of them is abnormal per se. So Many people who are completely healthy have many of these, but if you have too many of them, that increases the genetic risk burden, if you like, and increases the risk of developing MS. One of the most highly implicated and in the news relatively recently as an environmental risk factor is infection with Epstein-Barr virus. So this is a very common virus, and most people will have been exposed to it. It seems to be necessary for the development of MS, although clearly it's not sufficient because many more people have the virus but don't develop MS. Other risk factors include low levels of vitamin D, which we now understand is a clear risk factor, as well as smoking and being overweight. So those are, for the most part, the known risk factors, both on the environment side and on the genetic side. But we still don't fully understand why some people get MS and others do not.
So there are several things from what you said that people can do to reduce their risk. Maybe it's, keep healthy body weight, don't overeat, exercise, make sure your vitamin D levels are up either through sun or taking vitamin D, and then maybe migrating toward the equator.
Perhaps at a certain age, right? Below a certain age.
Yeah.
And then the smoking risk factor as well, which is actually true even for secondhand smoke is true.
Yes. Okay, then you mentioned, the two clinical manifestations of MS. There's this relapsing, remitting form, and then there's this less common, but I guess worse from the patient standpoint, the progressive disease, where they keep getting worse over time. All right, so I think we need to get into what's going on in terms of cells. So talk about axons and oligodendrocytes and myelination and the notion that the immune system's attacking, these cells, the myelin producing cells, or maybe even the neurons.
Right. No, that's exactly what we think is going on, that the central nervous system with its pathways, which involve the cells of the neurons, and then their axons, which carry the information to the next point, whether it's from the nervous system to the muscle or to another nerve in what's called a synapse, the connection between two neurons. Many pathways are wrapped in this fatty insulation, which is referred to as myelin, and myelin is made by these other cells called oligodendrocytes, and they are part of sort of the glial cellular matrix, if you like, of the central nervous system. And the oligodendrocytes wrap around axons and provide this insulation, which allows for the much greater efficiency of transmission. Yeah. Otherwise, we'd just be too slow and we wouldn't be able to escape from the saber-toothed tiger, right? And so the issue with MS is that at least part of it, the relapsing biology, involves these waves of immune cells that come from the outside, and around vessels, they create injury, and they create injury to the myelin, they create injury to the oligodendrocytes that make the myelin, and they can actually injure and even transect or create a break in the continuity of the axon. And these, of course, can contribute to symptoms because of insufficient transmission in different pathways. Many of the nervous system pathways are actually inhibitory, and if you injure them, you disinhibit something else. And so people who have parts, for instance, of the spinal cord or pathways to their legs affected, rather than having just weakness, they may have stiff weakness, spastic weakness, because there's the weakness, but there's also the increased stimulation, which is involuntary, and that creates issues with the transmission. So we think that the relapsing aspect of MS, again, involves these immune cells from the outside in, around vessels. But then, as you pointed out, there's another aspect to the injury of MS, which is referred to as the progressive MS biology. And this is something that appears now to involve some of the immune cells from the outside that set up shop. And now they basically are camping out within the central nervous system, and they're fostered there and continue to contribute to a percolating inflammation and injury. And that invokes some mechanisms that are referred to as degeneration. So there's a lot of interest in MS, both in inflammation and in degeneration. And in order to have treatments that I imagine we'll get into in a little bit more detail in a moment, we need to figure out treatments that on one hand can limit and ideally completely prevent these waves of inflammation from the outside in, as part of the relapse biology, but also deal with this inflammation or abnormal immune response and degeneration within the central nervous system, for which we may need to have different treatments or treatments that access different compartments.
Yeah, and looking through your trail of discovery in the scientific literature, at the time you started, my understanding is that the thinking was that the problem was mainly with one class of white blood cells called T cells. And then you and others, I guess, started looking at these other cells, the B cells. And you provide a lot of evidence that there's problems with the B cells too. So I guess if you could, kind of as simply as you can, describe what are T and B cells, and maybe talk about it in the context of COVID-19, because yeah, I mean, most people now know about infectious agents, and your body produces antibodies against molecules in the virus, and that's what the vaccines are doing. They're essentially mimicking, or they are molecules in the virus, and then your immune system responds. And in that case, that's good. You want your immune system to respond and clear out the virus. But in the case of MS, the immune system's it's, I guess, recognizing some molecule in the CNS as abnormal.
Yeah, so. Well, that's actually an excellent description, and you're absolutely right. I think all of us over the pandemic have had sort of basic training in immunology, by virtue of appreciating what the body tries to do against an infection, and then what we try to do with the vaccine to help the body mount an effective response. And the immune system is made, as you pointed out, by a variety of elements. Some of them are cellular elements, some of them are floating around, soluble elements, as we refer to them. Among the cellular elements, there are different types of immune cells, including T cells and B cells, and others. And the T cells have traditionally been viewed as kind of the conductors of the orchestra of the immune response. And they are involved in helping us recognize things that are foreign. And they, like B cells, have very specific receptors that are generally trained to do a pretty good job at distinguishing between everything that is foreign that we want to attack very efficiently, but not inappropriately attack self. And when someone has their immune system attack self, as you were referring to earlier, that's an autoimmune condition, meaning the system is attacking itself. And MS is considered a condition in which the immune system inadvertently is attacking parts of the central nervous system. And what we thought over many early years of beginning to understand the immunology of MS was that the main contributors to these attacks, to the relapses, were T cells of the immune system. And among them, there are T cells that help other cells. They're called T helper cells, and they were viewed as the most important ones. And by the way, one of the reasons for that being an attractive hypothesis was that there were several different animal models where people tried to create MS in animals. The most commonly used one by far has this long name, experimental autoimmune encephalomyelitis, or EAE. And all that means is that you're generating an immune response in the periphery of the animal, often by injecting it with a piece of the central nervous system, like a piece of myelin. That activates their T cells, especially their T helper cells, and they mobilize other cells and create attacks of inflammation and demyelination or injury to the same elements that I mentioned in the human condition. Now, EAE is not actually MS in animals, and the reality is that sometimes it has helped predict treatments that work, other times it hasn't. On occasion, it even said this should work in MS, and it actually made MS worse. So it is not MS in animals, but it is still a very helpful model for some of the principles of understanding how immune cells interact, how they can activate or regulate each other, how they can traffic to the central nervous system and participate in immune responses that cause injury. What we've learned in the human condition, and as you noted, partly our own work, but by many others in the field, is that the T cells do not work in isolation. And they really work in concert with other immune cell types and their subsets. And it's this kind of beautiful dance of cells shaping each other. And there are cells that are more pro-activation or inflammation, and others that are trying to ratchet it down. And this is normal. This is by design, because that very same response against the virus that you were referring to before, where you want the body to quickly recognize it and send its troops, in a sense, to get rid of the virus. And it does so by killing machinery, having very effective killing machinery, but you very much want to start ratcheting that back right away so that you get rid of the virus, but you don't injure self. And in autoimmune conditions, there's this spillover, maybe triggered by infectious exposure, but not necessarily. And it's where there's this kind of confusion where cells of your own immune system are attacking the myelin, the neurons, by mistake. And the treatments that we have and what we've learned through the treatments, which is a very important principle, where we've used the treatments to teach us more about MS. We've realized that by removing B cells selectively, you actually change very substantially new MS activity. You almost stop relapses completely in the great majority of patients, which surprised people. How could it that targeting B cells in a T cell condition would be so effective? Well, at least in part, it's because the B cells interact with the T cells, and when the B cells are present, they contribute to the abnormal T cell responses. So it's not about just T cells or just B cells. As I think of it, it's about the interactions between these cells. And we can actually think of every one of our existing therapies, as well as future therapies, based on how they may impact these cell-to-cell interactions in the periphery as they relate to the relapsing aspect of MS, but also in the central nervous system, because those immune cells are interacting with each other and with brain cells, and that is propagating the progressive aspect of MS.
So it's the B cells that are producing the antibodies, right? And so going back to COVID again, I guess, when you get, if you've had the virus or when you get the vaccine, you'll have these B cells, a specific set of B cells, called a clone actually. They're producing antibodies against some molecule, say the spike protein. And those can keep being produced for a long time and circulating in your blood, so that if you get infected again, they're recognizing it. Now, are there specific antibodies that are recognizing specific molecules in the myelin?
That's a great question. And, we've known again for a long time that there is an abnormal antibody response in people who live with MS. They have in their spinal fluid, and the great majority of people will eventually have this pattern of excess antibodies that look like they're produced within the central nervous system. They've been referred, as some people know, as oligoclonal bands, because they basically reflect certain clones of B cells and plasma cells that are the antibody-producing cells that seem to be propagated over time. But their target has been very difficult to sort out. And maybe some of them are relevant to myelin, others we don't really know. Maybe some of them cross-react with infectious agents. That's been one of the big mysteries. And what's really important to understand is that B cells contribute to a range of different autoimmune conditions, but they do so in different ways. And in some conditions, it is very clearly an antibody that B cells can abnormally produce that is creating the problem. And this is probably the case in the condition known as NMO, or neuromyelitis optica, where there are antibodies against the target on astrocytes that are very much involved in the problem. Another condition that is like MS is called MOG-associated disease, where there's an antibody against an antigen called MOG, which is a piece of myelin. And there again, we do think that that antibody is probably involved somehow in the actual injury process. In MS, it's still not entirely clear that that's the case. There are some hints that some antibodies in some people are probably involved in the MS process. For example, a recent study that suggested that certain cells may see EBV, Epstein-Barr virus, and as they react to it and produce antibodies against the virus, that in some people it may cross-react with a brain target, and maybe that's relevant to some people's MS activity. But it hasn't been found in the majority of people, and it is very possible that the antibodies are being produced, in at least much of the case, as an epiphenomena of the chronic activation of those B cells, but what they actually do that contributes to injury is some other property of the B cells. So B cells, yes, they can make antibodies, but they can do other things as well, and they can shape responses of other cells, including T cells. And we do think that it's those non-antibody dependent aspects of B cells that are more likely contributing at least to the relapsing MS biology.
And does the Epstein-Barr virus hide out in central nervous system similar to the herpes? Is that?
There's been a lot of interest, and some people have identified Epstein-Barr virus present in cells within the central nervous system of patients. Others have had a excuse me, others have had a harder time replicating that. So there's not 100% consensus on that. But EBV is very interesting because, as I mentioned earlier, there's no doubt, at least in my mind, I think the evidence is very compelling that EBV is necessary for the development of MS. Then the question is EBV also responsible for every MS attack? And then the third question is EBV also responsible for progressive MS? And there are ways in which one can think of EBV contributing throughout the spectrum of MS. It's possible. But the greatest evidence at the moment is EBV involved somehow in the early triggering of MS. And then it's plausible, but not yet entirely proven, as to whether all MS attacks involve, for instance, EBV-infected B cells that may trigger T cells that may then respond to the myelin, either through the T cell response or the antibody response. Those are plausible mechanisms, but not proven. And finally, to your point, is EBV present within the central nervous system, where its biology is somehow percolating the inflammation there and contributing to the ongoing injury of progressive MS? Again, possible, but not entirely proven. And several groups, including ours, are trying to better understand this by doing trials, clinical trials with treatments that are designed to actually test these questions. In other words, we're going to try to get rid, for example, of EBV-infected cells, and then see whether that actually makes a difference both to the relapsing and progressive MS experience.
Two questions on this Epstein-Barr virus. Does it infect oligodendrocytes? One can culture oligodendrocytes, for example, and I guess see if it will infect them. That's the first question. And the second is, you mentioned that EAE is not an ideal model for MS, this mouse model. Will the Epstein-Barr virus infect mice or not?
So it turns out 2 points. One is that EBV does not readily infect oligodendrocytes or oligodendrocyte precursor cells. It infects some B cells, and it affects epithelial cells. So it's commonly involved in when people get EBV, there's shedding of EBV in the saliva from epithelial cells in the oropharynx, in the mouth and the back of the throat. Then the issue of whether one can think of EBV as a therapeutic target. I think that there, again, we will learn a great deal as we intervene with EBV. Our challenge in animals has been that the typical animals we use don't get readily infected with EBV per se. It's not really a virus that infects mice, for example, in the same way. There are other gamma herpes virus family members that have been used to successfully produce infection in animals, and people use them in part and with increasing interest recently to try to explore how EBV may be relevant to MS. The animal models that we have to date, in my mind, continue to be important, but we need to understand what they are useful for and how to interpret them in the most meaningful way, so that we don't ask the animal model to be more than it actually is, but learn those things that it is best suited to teach us.
Yeah. Okay. So it's not clear then whether the primary initiating factor for MS is a problem with the immune system, or there's a problem in the CNS cells that then lead to this abnormal response of the immune system. Is that right? It's not
well
clear.
So over the years, there was a lot of weight placed on the immune system driving MS, and again, animal models like EAE made us all think that yes, this must be what's going on. And almost certainly the immune system plays a key role, probably a primary role, although what starts it all is a good question. And one of the ways to ask about what starts it all is to look at the genetics. And it's interesting that those 250 some genetic variants that I mentioned to you now have been implicated as risk factors for MS. The great majority of them seem to be genes that affect immune system cells, but not all of them. And some of them appear to be relevant to brain cells. So there probably is an element of risk at least from my perspective, that may involve the target organ. And it's a fascinating question to ask about why are the target organs involved in different human autoimmune diseases different.
Yeah.
Most people who have an autoimmune disease have an autoimmune condition that affects certain organs.
Yeah.
In the context of MS, it's really the central nervous system. In other conditions, it may be several, but it's not every organ in the body. And that probably reflects some contribution of genetics and environment that determine what the more susceptible target organ actually is. And so as we look through these genetic variants with a different optic, if you like, not as immunologists, but as neurobiologists, we begin to realize that there may be some very interesting aspects that contribute to susceptibility at the level of the central nervous system itself, so that there may be people who have all of the risk factors on the immune system side, but somehow their central nervous system is better protected and they will not run into a condition like MS. Others may have relatively milder problems on the immune system side, but bigger problems on the central nervous system side, which for that individual is enough to have MS.
Now, you've been involved in many, many clinical trials in MS over the years and even maybe decades by now. So why don't you kind of give a historical perspective on the treatments? We're going to finish up, talking about treatments, and there's a lot been done, a lot going on. So you want to kind of, I guess, go back to, I guess I'm not sure, I'm not an expert on this, but I know beta interferon was given at one point.
That's right.
Yeah.
So the beta interferon, at the time, beta-seron or beta-ferron, was the first approved immune modulating treatment for multiple sclerosis. And it was shown, and it was actually partly through the introduction of the MRI as a way of looking to see how people did over time, because it turns out that there's much more activity of these relapses under the surface than above the surface. And that's true both for the relapsing aspect of MS and the progressive aspect of MS. And I mentioned this, it's relevant to how our clinical trials have evolved, because we've been trying to identify additional ways to measure the consequence of these treatments in terms of both the relapsing biology and the progressive biology. And one of the impediments over the years had been having to wait till people actually experience more clinical attacks or clinical progression. And to do that, and to be able to show that a drug is better than something else, you needed many, many people to be followed for very long periods of time, making these studies also extremely expensive. Beta-seron was the first one that was approved in the world of MS. It was extremely exciting, and it was shown using the MRI as well as measuring clinical relapses that it decreased new MS relapsing activity by about 30%. Now, 30% doesn't sound like a lot, but at the time it was unbelievably exciting because it really was this transitioning of MS into a treatable condition. It required injections under the skin 3 times a week. It had a bunch of side effects associated, but it was a welcome transformation in the MS field. Other injectable therapies evolved with glutamer acetate as well as other interferons. They all require, again, injections under the skin or into the muscle, different frequencies and different side effect profiles, all of which were modestly limiting new MS activity. And then we saw several oral pill form medications for MS, some of which now were easier to take and more effective. So instead of having an average ability to limit relapses by 30%, they might do it by 40, 50, even 60%. And then came the monoclonal antibodies, which are much more targeted in their approach, and then have the benefit in principle of having lesser side effects. Although all of our treatments have over time been found to have some issues with either tolerability or even safety, not very big safety concerns in terms of high frequency, but sometimes a low frequency concern of something very serious, such as an infection of the brain called PML, or progressive multifocal leukoencephalopathy. And one of the other challenges about the evolution of therapeutics is that you end up realizing that a treatment may be very good, but may have some risks. And if only you could understand who is at risk and who is not, you could then offer this treatment as an effective, well-tolerated, and very safe treatment to many, and spare the others and think of a different treatment for them. So this is another theme that I think is running through the development of our clinical trials in MS. One, again, being how to measure things, but the other also how to monitor things and how to try to identify who may be more at risk or who may be most likely to benefit. Because if you're most likely to benefit, maybe you're also willing to take a little bit more of a risk. And this is kind of the move towards a more personalized approach to understanding people's MS. We now have several treatments that are very, very effective, including the B-cell targeting therapies that are very effective at limiting relapses. And we understand that if we wait and use them later, people don't catch up. So there's an imperative to diagnose early and have a discussion at least about high efficacy therapy early on, because you won't catch up. And while our treatments currently are doing a pretty good job, and we should be able to find one that works for just about every individual to very substantially limit their relapses. So this is a major, major improvement. We don't really have very effective treatments for the progressive aspect of MS.
Interesting.
And the other that we still as a field are trying very hard, and it's moving slowly, is how to better understand how to fix things, protect things, or fix things that are injured so that we can actually repair and provide some important recovery to people who already have deficits from their MS.
And so when you do the imaging, do the lesions in the brain look the same with the relapsing remitting versus the progressive form?
Well, that's a really interesting question. The lesions that we see pretty readily on the typical MRI are really more a reflection of the consequences of the relapsing biology. It is focal, meaning it is in a particular place, and it is typically around a vessel, and we can see those in the brain quite readily. It's a little harder to see them in the spinal cord, but they can be seen there as well. And it makes it relatively easy to look for new ones and count them, and then be able to comment about a treatment being able to effectively decrease the development of these new lesions. The progressive aspect of MS happens in somewhat different places, is more diffuse, not quite as focal, and it has not been as easy for a number of reasons to visualize with the MRIs, certainly not in the regular clinical MRIs. It contributes over time to loss of tissue. The combination of the relapsing biology injury and the progressive biology injury contributes to atrophy, which is some loss of tissue over time. And one can look at that over time in populations and clinical trials, but measuring that only tells you that there's ongoing injury. It doesn't tell you what the source or the cause of that injury is. We've also, in the field of MS, become excited about the prospect of being able to develop biomarkers, biological measures, in addition to the MRI, which is a form of biological measure, but in addition to the MRI, to try to look at things that may be soluble products that identify the biology of MS under the surface, so we can pick it up even more sensitively without having to wait for something bad to happen. And can we look at those measurements to predict who is at greater risk, who is more likely to have activity? Can we use that, of course, to try to implement into the care of patients? And we're not quite there yet, but I think we're getting close. And can we use these types of measures also to enrich clinical trials to be most informative? Because, for example, I mentioned a major unmet need is progressive MS. There are people who progress very slowly. And if you have a whole clinical trial with people who progress very slowly, it becomes extremely difficult to appreciate a benefit of a treatment, and the trial may be negative. You know, patients are often frustrated, and I think rightly so, when a clinical trial doesn't include them because they're not eligible. And the reason for that is that there are pretty strict inclusion and exclusion criteria, and some are designed to keep people safe in the clinical trial. Others, though, are designed to try to make sure that the clinical trial has the best chance of showing a benefit if a benefit exists. If all the people in the trial are not going to be informative, this will be a failed trial, and that drug will be dropped and will never help anybody. So that's one of the challenges of clinical trials, that it becomes a microcosm of that trial. And then the question is, will the result generalize more broadly to other people?
In the description section for these podcasts online, I'm going to put in some links to some of your review articles and relevant to the treatment one. You had a nice one in Nature Reviews Neurology fairly recently put in there. And one thing I noticed in there, so you mentioned the monoclonal antibodies, which I assume they're targeting some protein in a particular type of T or B cell and trying to damp down the immune response. But I saw other treatments in there too that seemingly don't seem to be immune system specific, like alpha lipoic acid. Now, is that something that really helps? And
well, so it's early days for that and several other molecules that are described in that review, but it is clear that if we only affect the immune system outside of the nervous system, we may be very effective at limiting relapses, but that need not translate into effectively limiting progressive MS or repairing the tissue. And so we need to do more than just that. And you could imagine one prospect is to have the same molecule, the same treatment accomplish both. And that may be the case. There's a relatively new class of medicines that are now already in advanced clinical trials. Those are called the BTK or Bruton tyrosine kinase inhibitors. And they work on modulating immune responses of both B cells and cells called myeloid cells in the periphery. But if they get into the central nervous system, they have a chance of limiting this abnormal percolating activation of B cells, but also of brain cells called microglia that we think may be quite relevant to progressive MS. So there's a prospect of a treatment, if it works both in the periphery and can access the central nervous system, may accomplish more than just limiting relapses. Alpha lipoic acid is an example of a treatment that we think has a chance of getting into the central nervous system, and there may have an effect on some of the brain cells and may then shape not just the immune response that we're trying to acquiesce, but also promote an environment that may be more supportive of repair. And that, of course, and with other medications, interventions in sort of those classes, as well as other interventions. You mentioned exercise before. There's a lot of interest in whether one can engage in exercise in ways that may promote repair in the central nervous system. A lot of discussion around the diet and can that work? And I think that we still have a lot to learn, and that these are exciting prospects. We need to figure out how to study them and studying them well so that we can provide, compelling evidence that it does or doesn't work. If it does, that's great. If it doesn't, then we move on to other things.
We did a lot of work on intermittent fasting and its effects on the nervous system in various models. We didn't do it in the EAA model, but actually there's other people that have since done it. But the idea would just, do the healthy things, exercise, keep your weight down. You mentioned microglia, and so I have a niece that has Crohn's disease, and she gets, which I guess is what a common treatment now is, antibodies against it's called a pro-inflammatory cytokine called TNF, or tumor necrosis factor. And I know microglia produce a lot of TNF. Have there been trials of TNF antibodies in MS?
There have. Both of the antibodies as well as the soluble receptor, which mops up TNF and achieves a similar thing of decreasing the transmission of TNF signaling. And actually that harkens back to my earlier comment. They did this in EAE because in EAE, it certainly looked like TNF was a bad player and it helped EAE. It prevented bad EAE. But when introduced into early trials of individuals with MS, it actually caused new increased MS disease activity.
Interesting.
And so it's one of those molecules that worked to decrease EAE, but clearly made MS worse. And there are several of those. And they teach us something extremely important beyond the realization that EAE is not MS in animals. It teaches us something very important, that modulating that pathway can change the face of central nervous system inflammation. So it is somehow involved in these cascades, or can be involved, but it's doing it in the wrong direction. Yeah. And why that is and what we can learn from it to do differently is very important.
In the early 1990s, we were doing a lot of work looking at how different neurotrophic factors, proteins that promote the growth and survival of neurons, may protect neurons against dysfunction and degeneration, and models that are relevant to Alzheimer's, Parkinson's, and so on. And then we started looking at cytokines. A lot of this was in cell culture. We just have cultured neurons from the brain. We treat them with a growth factor or a cytokine, then expose them, say, to the amyloid protein that accumulates in the brain, or to what we call excitotoxins that overexcite the neurons and cause their damage. And so I had this postdoc, he'd done some work with the growth factors showing they protect the neurons from being damaged in these models. And then I said, well, we should look at some of the pro-inflammatory cytokines, thinking it's going to make it worse. And actually, we found TNF was neuroprotective.
Yeah.
And that it acts through a certain gene expression that promotes the survival of the neurons. And we had a lot of trouble getting that paper published back in the early 90s.
But it's very clear, you're absolutely right. It's very clear that there are partly these what we call pleiotropic effects, meaning many different effects.
Yeah.
The same molecule can have a very different effect in a different context. And part of it has to do with the cells that may be exposed to this factor may have different receptors, and different receptors to the same molecule can have very different functions, including, as you point out, something that can be good versus bad in a particular context. It's true for TNF. It's true for another cytokine called IL-1 beta that can have pro-inflammatory, potentially damaging effects in the context of CNS inflammation, but also is a molecule that seems very relevant for aspects of repair, for the neurobiology of remyelination. And I think one thing that I'm reminded of again and again, as someone who straddles the field of immunology and neurobiology, is that in many ways, we have artificially carved out the world of health and ill health by giving different fields names and saying this tissue or these molecules or this therapeutic belongs to us versus, and it turns out that there are clearly molecules that immunologists think they own, but are expressed and used by brain cells.
Yeah.
And there are clearly molecules that neurobiologists think they own, such as growth factors that immune cells actually use. So for instance, there is a very clear subset of memory B cells that express nerve growth factor receptor and secrete nerve growth factor as a way of keeping themselves alive over time. That's one of the mechanisms by which we have long-lived memory of the immune system, because of these tricks that are really a reflection that there are many, many aspects of overlap. And we need to keep a very open mind as to,
I've thought about, Amit, I've thought about this from an evolutionary perspective because, immunologists discovered a lot of these molecules and signaling pathways for a simple reason. You can just take blood from a person and isolate the blood cells.
Yep.
And then, so we start seeing TNF affects neurons. And then I got thinking, my understanding is during evolution, organisms had nervous systems before they had what's called the adaptive immune system, where you've got these circulating white blood cells. So I'm thinking from an evolutionary perspective, maybe these signaling pathways, some of them were first in nervous systems and then later, rather than the reverse.
Yeah, and then there's co-evolution, and I think that's exactly right. No single system has a monopoly over how biology was either created or evolved, or a combination of the two. And I think that it's been fascinating to see. It also means, by the way, that when we develop an intervention, a treatment that is meant to target what we think is a molecule on the immune system, we have to remember that it may have effects on other tissues, including the nervous system, and vice versa. So there's how do we leverage this to benefit as opposed to create an adverse type of side effect, right?
Yeah. Okay. To end with, what's your kind of outlook for MS? What are the most exciting things going on now in MS research?
Well, I'll start maybe, Mark, by just mentioning that I think that this is a new age of MS care. When people are diagnosed nowadays, one of the very first things that I suggest is that, this is a diagnosis that you may not have invited, but you have. And important to understand that we now think of MS as the new MS. Because being diagnosed with MS today is very, very different from having been diagnosed with it years ago, certainly decades ago, but even 5 years ago. We now, our job, and we should be successful, is to keep you as good as you are, certainly in terms of control of relapses, and as time goes on in additional ways as well. So I think the prospect of MS, living with MS, is very, very different from what it was. And that's an important, I think, very valuable message for people to hear. Also, that there are many choices, and that what people used to be confronted with, it was sort of, this is the most important decision of your life. Well, it's not an unimportant decision as to what you may start, but whatever you start with will be monitored and can change if needed, either because of tolerability or safety, or because of insufficient efficacy for your MS. And there are many things to choose from. So whatever decision you make is the best that you could have made at that time, but is not a marriage for life, is subject for monitoring and change. And that also unloads those initial considerations, I think, in a very important way. To the field of therapeutics, per se, I think that we, again, have not quite finished everything there is to be done therapeutically in terms of relapsing MS. For example, while we now have treatments that can really stop relapses in many individuals, and treatments that are quite well tolerated and surprisingly safe, nonetheless, we don't think of them as being safe forever. And a very important question is, when do we stop? And so there's a whole field in the MS area now of thinking of at what point may a certain treatment that may have been very helpful over the years no longer be helpful. Because of course, MS attacks, for instance, they tend to diminish with time anyway. And eventually people living with MS stop having those attacks without any treatment. So if they're not having an attack while they're on a treatment that they've been on for a while, are they not having an attack because of the treatment still helping, or because they're no longer going to have an attack? And that's a challenging question, but there are now trials looking at that in particular. The other prospect is to treat with something and then be able to stop, and for people not to have MS activity come back, even in the earlier stages of MS. And that, for an immunologist, is essentially what we refer to in the medical sort of parlance as restoring immune tolerance. Because if you have an autoimmune disease, it means that somehow you lost tolerance. Your immune system is not entirely tolerating yourself. But can we restore immune tolerance? And we have the opportunity to learn that, I think, as part of the autoimmune field more generally. And some of the studies that we've been very excited about and involved in are, what can my condition, so to speak, teach you about yours, and vice versa? So how do we work with people in the diabetes field, in the inflammatory bowel disease field, et cetera, to do things in a coordinated way and figure out what one condition can teach you about the other? So there's a lot of opportunity there. There is clearly opportunity to do better than just a modest benefit on trajectories of progression. And so there are several treatments and pipelines in both advanced and early phase. And I hope that if we talk again in 10 years, we'll be able to say that we've licked progressive disease in a similar way to what we've been able to do with relapsing disease. I think that the issue of fixing things that are injured, that's going to be more challenging. Probably more than a decade, but perhaps not much more. But all the more reason that we need to do our very best to prevent damage with whatever ways we can, until such a time that it's easier to fix damage that already occurs.
Yeah, very exciting area. I appreciate you taking the time for this. I know you're extremely busy, so
pleasure.
Appreciate it. And I wish you happy holidays and happy New Year.
Thanks, Mark. Same to you. Thanks for having me.
Take care. Bye.